So good afternoon, everyone. My name is SANJUAN HIDALGO, and today I'm going to present my work, CRISPR-Cas9 mediated edition of KRAS driver mutation in lung adenocarcinoma. So first, let me tell you what is lung cancer. Lung cancer is, along with breast cancer, the most incident cancer worldwide according to Global Cancer Observatory data of 2018, and at the same time it is by far the cancer that has the highest mortality. So lung cancer can be further subdivided into different groups. Small cell lung cancer with around 20% of the cases, and non-small cell lung cancer with around 80 or 85% of the cases. An important feature of non-small cell lung cancer is that it has been observed that the oncogene KRAS is mutated in up to 30% of this tumor. This is such an important percentage, and this has very important biological significance. So, what is the importance of KRAS mutation in lung cancer, in lung adenocarcinoma? 90% of mutations have been reported to be localized in a particular exon of the gene, the exon 2, and particularly we are interested in these two mutations, the G12C and the G12D mutations. They are missense mutations. This means that they change the instruction for just one amino acid. Okay, so they provide a new instruction for the synthesis for a KRAS mutant protein, hyperreactive mutant protein that is stays bounded to GTP and is no longer able to return to its inactive state. So now the KRAS mutant protein is able to transduce signal to the nucleus, proliferation signals, in order to activate proliferation genes, and this eventually is able to initiate tumor development. So for this reason, these mutations are called driver mutation, because only with this mutation on KRAS is enough to initiate tumor development. So even when there have been several inhibitors developed to specifically target a mutant KRAS forms, some of them have been shown to present toxicity on the patients and also limited efficiency, even when they have reached phase 3 of the clinical trials. And also, in some cases, it has been reported resistance of the tumor for these inhibitors. For this reason, it is necessary to develop a new therapeutic approaches. And the goal of our group is to implement the CRISPR-Cas9 system in order to selectively target KRAS mutant alleles in order to develop a new therapeutic approach. For this reason, we want to combine the CRISPR components. In this case, we use enhanced version of Cas9, which has a reduced off-target activity, and we mix this Cas9 with the guide RNAs targeting the mutations. So when we expect that when we introduce our CRISPR tools into a KRAS wild-type cells, we should see no effect on cell proliferation, but when we introduce our CRISPR tool into KRAS mutated cells, we should see addition of KRAS and eventually cell death and tumor regression. And for this, we have developed two different approaches. The first one, an in vitro approach, can be subdivided into different sub-approaches. First is the transfection with the ribonucleoprotein complexes. This is a very simple approach consisting on mixing the purified Cas9 in a tube with the guide RNAs in order to form these complexes. And we use these complexes in order to transfect the cells, and we check cell viability after 7 days. Also, as we wanted to reach better addition efficiencies, we take advantage of adenovirus infections in order to check cell viability in 2D culture and also in 3D culture. And this is important, the 3D culture format, as it resembles to more or less or more than the 2D culture to the tumor environment. We also check the inhibition of spheroid formation that are, let's say, small tumors. The other approach for this project is an in vivo approach, in which we use the same adenovirus in order to infect the cells, and we then transplant these cells into immunocompromised mice, and we evaluate the ability of these transduced cells to form tumors in vivo. So regarding the specificity of the guide RNAs, when we introduce our CRISPR tools into wild-type cells, we don't see any pattern of addition. We evaluate this addition with this T-cell mutation detection assay, but when we introduce our CRISPR tools into mutated cells, we see the expected pattern of addition. The same happens with the G12D mutation. We don't see any pattern of addition, but we obtain it when introducing the CRISPR components into mutated cells. Also, we have checked loss of cell viability in this set of cell lines carrying the two mutations. And importantly, we don't see any effect on cell viability in these cells that have the wild-type KRAS, as we are not editing KRAS. We also have checked this behavior by measuring through the colony formation assay, and we can see that we have less colonies in those cells which have been edited, and the exact number of colonies appear in the control cells. Also, in the adenovirus part, we have checked that, well, these are very fresh and preliminary results. We have checked that we are able to reduce cell viability up to the half, more or less, and this reduction in cell viability is reduced up to 70%. Also, we can see here this spheroid formation assay. We can see here in the controls little tumors, let's say that appear in the control, but nothing can be seen in the treated cells. Also, for the wild-type cells, as I said, these are very fresh and preliminary results. We don't see any effect on cell viability in both 2D and 3D cultures. Also, in the in vivo approach, we have checked that when infecting the cells with our virus, we are able to reduce tumor volume in these two cell lines. In these two cell lines carrying the two mutations. Also, these images could be disgusting, maybe for someone else, but I wanted to show you the reality behind the graphs. And for example, here we have a very nice reduction of the tumors. Also here we can see a loss of the KRAS mutant protein, and also this mouse is only one of the cases in which we even are not able to find the Cas9 tumors. And finally, as a conclusion and take-home messages, I would like to say that CRISPR-Cas9 system can be implemented to selectively target KRAS driver mutation without affecting the wild-type allele. The targeting driver mutation of KRAS suppresses tumor phenotype at in vitro and in vivo models. And as a future perspectives, CRISPR-Cas technology has opened the door to be an alternative approach for lung cancer and even for any other cancer, and can be implemented for other key genes such as EGFR. And finally, I would like to thank my wonderful group, and it's increasing number of people, also the funding agents. And I would like to thank the scientific committee for selecting my work for oral communication.